His honor include National Science Foundation Career Reward, and he's also a member of the American Institute of Medical and Biological Engineers. So we are really excited to have Professor Stephen Chase today with us at our talks, and I will hand over to Professor Stephen Chase if Vikash have something to say before that. Vikash, do you have something to say?
Let's begin.
All right. Thank you so much, Håkan, for the intro and for the invitation. I'm excited to share my work today. So let me share screen. There we go.
Just before the for all the audience, feel free to jump into the presentation. So we are like discussion mode. So people are feel free to jump in. Don't need to wait to the end. Yeah.
Oh, it stopped sharing. I am sharing screen. Are you seeing the correct screen?
Yes, I've seen neural mechanism of
Perfect. All right.
Yeah.
Excellent. So yes, super happy to share my work with you all today. So I've been interested in volitional motor control for a really long time, and a lot of my work is in brain-computer interface design. But I want to share with you today some work that started about 8 years ago on some non-volitional factors that influence how we move. And so to start with that, I want to start with a hypothetical. Imagine a scenario where I have you play a little game where you putt a golf ball into a cup. And we can set it up so that when you can putt successfully into the cup, maybe 70% of the time or something. And then imagine I told you, I'll give you $1 if you get the ball into the cup. Chances are you would do slightly better. And then if I told you, you know what, I'll give you $10 each time you get that ball into the cup, chances are you do a little bit better yet. And then, and again, this is a hypothetical, but I could tell you, I'll give you $200,000 each time you get that ball into the cup. Chances are you'd actually do worse. So this phenomenon is known as choking under pressure, and it's super familiar from all aspects of sports. There's usually, any one of us has some known event in time where their favorite athlete has just failed at a critical moment. But it's also well studied in the lab. For some reason, humans, at the most critical times when it's important to succeed well, we tend to underperform. And that's the part that a lot of people focus on. But I would say that this relationship between motivation and performance has two kind of equally important and interesting features. The first is that we get performance improvements with increases in motivation. And so that's interesting to me because it seems like, shouldn't we just be performing as well as we can all of the time? Why should we have these performance increases? Why should the system have a capacity that's not used? And second, we have this choking under pressure effect where the system fails us right when we want to use it the most. And one of the major questions that's been driving my work for a little while is what neural mechanisms link motivation to movement? And if we want to get at neural mechanisms, how the brain is mediating these things, we need to go to animal models, all right? And the first question we had when addressing this work is, do animals show the same kind of motivation performance relationships that humans do? Will animals even choke under pressure? And so to answer this question, we designed a task that could be performed by monkeys. So we study rhesus macaques. And the task that they do is a little video game. And it's a movement task where they hold their arm up, and we've got a little marker on the back of the arm that emits in the infrared and can be tracked by a bank of cameras. And that marker controls a cursor on the computer screen. And the task is as follows. The monkey needs to acquire a center target. So a target shows up on the screen and he moves his cursor to overlap with it. And then we show a target on the screen. This is a target he will have to reach to, but he's not allowed to reach to it yet. We have a delay period of some random length of time. And at some point, we're going to give him the go cue. And at that point, the monkey needs to execute a fast and accurate reach to hit the target. And if they do so, they get a reward, drop of juice. Now, the key here is that the target tells the animal how much reward is on offer. All right, so in some monkeys, it was the color of targets and of the target. In some monkeys, it was the shape inscribed in the target. So the monkey has knowledge of what the reward is going to be on this particular trial. And we varied that reward from trial to trial. So on some trials, on almost a third of the trials, it was a relatively small amount, 0.1 milliliters. On roughly a third of trials, it was a medium amount, 0.2 milliliters. On roughly a third, it was a large amount, 0.3 milliliters. But here, on 5% of trials, we gave them a jackpot reward on offer, which was 10 times the size of the medium cube. And our question was, for this task, the task requirements are the same across all 4 reward conditions. The question was, how would the monkey succeed as a function of reward? And so here I'm going to show you the success rate as a function of each of these reward conditions. And we titrated the task. It had to be both fast and accurate. And we titrated it so that he was about 70% successful on average. So for the small reward conditions, he succeeded around 60% of the time across, this was 2,000 trials in this case. And then for medium and large conditions, we saw an improvement in performance. Performance improved to roughly 85%. Right? So we see that we got the same increase in performance from small to large rewards. What happens at jackpot rewards, and indeed monkeys do choke under pressure, where we get this dramatic decrease in success rate from 85% down to about 65% in this animal. Now, turns out this is true for every animal that we've studied so far. So here's 5 animals in the same task, and it's a very robust effect. Each of these animals shows a performance increase between small and large rewards, and a performance decrement between large and jackpot rewards. This turns out to be a remarkably reliable effect. So we get a lot of questions, well, what about the second day, the third day? Do they get used to it? Do they just, start to understand it? And the answer is no. So I'm showing here each of these lines is the success rate curve for a single day for each of these monkeys, shaded from day 1 until the last day that we studied them. And each day for each monkey shows a performance decrement with the exception of one curve in monkey F, which didn't. So the effect is very reliable across days. We looked at the first half of the day and the last half of the day, wondering maybe, they get tired of the juice, they get satiated, maybe the choking under pressure effect goes away there. Nope, it turns out that we get choking under pressure for the first half and the second half very reliably. So this effect is reliable within the session. We looked as a function of target direction. We had monkeys make reaches in each of 8 different directions, and monkeys showed this performance curve for each of those directions. So it's reliable across the different movement types that we studied. Now remember, monkeys had to wait for a certain period of time before that go cue came, where they got to think about the reward that was on offer. And that delay period varied over some random period of time. So we looked as a function of the delay length in milliseconds here, from our shortest delays of 250 milliseconds to our longest of 950 milliseconds. And they showed reliably decrements, choking under pressure for all delay conditions. So this was a reliable effect across delay durations. And then finally, we had monkeys do this task in bursts. We'd study it for 2 weeks. We go and do some other things. And in one animal, we studied it across a span of 6 months. And for each time we went back to this task over the course of 6 months, the animal showed a reliable effect where we get performance improvements from small to large, and again, performance decrements from large to jackpot rewards. So it was reliable across long time periods as well. And the overall conclusion here is that monkeys choke under pressure extremely reliable, possibly more reliably than humans do. All right. So now the question is, we see these success rate changes between small and large reward and between large and jackpot rewards. What's actually happening in the kinematics that lead to these success rate changes? So we broke down the reach itself. And so here we're going to talk just about the reach component of this task. And there's really 3 ways that this reach can end. First, the animal can land on target within the time window that we allow and hold on target correctly, and that's a reach success. The next thing that could happen is that the animal moves too slowly and runs out of time, or he makes a reach that undershoots and doesn't have time to correct. And so that's an undershoot, and that's one type of failure. And the other type of failure that we see is the opposite of the undershoot. It's the overshoot. It's where the monkey blows through the target, lands too far. Now there's another where the monkey could reach in the wrong direction entirely. We never see that kind of error. So these are really the three errors that we see. We see reach successes, or three types of ways the reach ends. We see reach successes, undershoots, and overshoots. And the question is, what's happening as a function of reward to these different outcomes? So if we look at the large reward, this is where the animal is most successful. We see that primarily it's reach success. In the light gray, we get a small amount of undershoots, and in the dark gray, we get a small amount of overshoots, and they're roughly balanced. Now, when we look at what happens with medium and small rewards, we see an increase in both types of errors, but an increase in undershoot and an increase in overshoot behavior. Now, another question is what happens with jackpot rewards? And for jackpot rewards, we see consistently an increase just in the undershoot behavior. So monkeys, this monkey especially, undershoot more in jackpot reward conditions. So again, we checked all of our animals to see if this was a consistent effect, and in fact it was. All error types decrease between small and large reward. Undershoot errors increase between large rewards and jackpot rewards. All right, so as this just restates what I said, is reward increase from small to large, both error types decrease. For jackpot rewards, animals tend to undershoot. They move too cautiously. All right, so I just want to summarize the behavioral part of this talk. Motivation modulates behavior in monkeys just as it does in humans, including this well-known choking under pressure effect, where for jackpot rewards, we underperform when it matters most. Performance modulation by motivation is incredibly reliable. Under each and every condition we've looked at it, we see that it exists. And all animals choke due to undershooting the target for the highest levels of reward. They tended to move too cautiously and run out of time. All right. So now the question is, what's happening in neural activity? What is it about reward? How reward modulates neural activity that's causing these kinds of performance improvements from small to large, and then performance decrements from large to jackpot? So to do this, we need to look in the brain, and we decided to focus our initial studies on the primary motor cortex. The primary motor cortex, located roughly about here, is the source from cortex that sends direct projection down the spinal cord to help you exert control over muscles. So it's the last cortical stop for volitional motor control. And it's a convergence point for reach information, including the goal, the location of the target, as well as the posture of the arm. All of these signals come together so that you can create that volitional motor command. And it also receives inputs about motivation and arousal both from brainstem circuitry sources like the basal ganglia and the locus coeruleus, as well as from prefrontal cortex. So it's a convergent point, both for motor command information and for signals that carry motivational value. All right. And so we record using microelectrode Utah arrays. So these are small chips of silicon that have recording electrode shanks that can record individual action potentials from up to hundreds of neurons. And so we implant these arrays in primary motor cortex and in dorsal premotor cortex, and we record from dozens of neurons in any given session. And so what I'm indicating here, each row is a different neuron, and each tick mark is the action potential that neuron releases at that time. And the first thing I'm going to do is show you some single neuron results. What's happening in single neurons in primary motor cortex when you are experiencing different types of rewards. And so what we're going to do is just average the firing rate across reward conditions, so the number of action potentials per second. And I'm going to show you here 3 neurons. And this is a curve as a function of time of the average firing rate. The first dot here is when we show the target that indicates the reward that the animal will be receiving. And the last one here is the go cue. So this is aligned to when we gave them the go cue. And so I'm showing in red here, this is the average firing rate for the small reward condition. And to compare, we can look at the curves for the medium reward condition and the large reward condition. And what we see is that we get these modulations of firing rate in a large number of our cells. Some cells increase their firing rate with reward. So you can see that the yellow curve is above the red and the blue curve is above the yellow. Some cells, like neuron 3 here, decrease their firing rate with reward. but we see this consistent modulation of firing rate with reward. Now, one of the questions we had is what would happen under this jackpot reward condition? And one of the hypotheses we had is that the reason you tend to fail under these jackpot reward conditions is maybe there's an insufficient drive to motor cortex, where these motivational signals, for whatever reason, aren't as large as they should be under jackpot reward conditions. Maybe you stress out, maybe you get nervous, and so maybe that motivational drive isn't there. So one question was, do jackpot rewards lead to this sort of insufficient drive? And we see no evidence of that. What we see is that jackpot rewards continue that trend that we see from small to large. So if reward rates go up between small and large, jackpot conditions cause those firing rates to go up even more. If firing rates go down from small to large, jackpot conditions cause them to go down even more. So the response to the jackpot reward is monotonic. All right. So that was our single neuron responses, but we're recording hundreds of these neurons. And in fact, the motor command is really composed of thousands of neurons working together to drive the muscles. And so we need techniques to analyze all of this data simultaneously. So I'm going to walk through how we do that. And the way we do that is I'm going to, again, just consider 3 neurons for a moment. And what we do is we take a small section of time, maybe it's 100 milliseconds, and we bin it and we count the action potentials in that section. And we look at the firing rate in this bin of neural activity. And what we can do is we can plot that in a space where each axis is the firing rate of one of those neurons. So for these 3 neurons, it's a 3-dimensional space, and the simultaneous firing of all 3 neurons can be considered a point in this space. That's a neural activity pattern. And as you can slide time along, you get different neural activity patterns in this space. Now, of course, I'm only looking at 3 of the hundreds of neurons we're recording. We're recording a lot more neurons. And really, the trick here is that instead of looking at these neural activity patterns in a 3-dimensional space, we actually look at them in an n-dimensional space, where n is the number of neurons we're recording. So often it's sort of a 90-dimensional space. We're recording 90 neurons. So we can visualize 3 dimensions at a time, but we can mathematically operate on all 90 to 100 dimensions. And each point, again, represents the simultaneous firing of all of the recorded neurons for that period of time. All right. So one of the things we did was we looked right before go cue, when the monkey's waiting to make that movement and they're ready to execute it. And we looked at, instead of just one neuron at a time, we combined the activity of all of our recorded neurons at the same time in this high-dimensional neural space. And the first thing we did was we found an axis in this space that represented the greatest variance that was explained by reward. All right, so we saw one axis. We used principal components analysis to find a single axis that explained the largest amount of variance in this space of the reward conditions. All right, we call this the reward axis. And it's a linear combination of neural activity. And so we can plot the projection along that reward axis for each individual trial as a function of the reward condition. So red here are the individual trials for the small reward condition, yellow for the medium reward condition, blue for the large, and black for the jackpot. And again, we see that the primary response to reward here in motor cortex is monotonic, right? We get monotonic increases along this reward axis projection. It's increases in some firing rates and decreases in the other, but along this projection, monotonic change in projection as a function of reward. All right. So we have a monotonic change in firing rate with reward, but we have this non-monotonic behavioral change. We know success rates go up and then they come back down. So clearly this monotonic change isn't explaining that non-monotonic behavior. So what else might be going on? So the next thing we looked at is how this reward information interacted with the motor command signals. And so to do this, again, we can find combinations of neural activity, linear combinations. But in this case, we're looking for linear combination of activity that separate the target conditions, so the actual motor commands themselves, so different directions. And we can look for these 3 example neurons. Here I'm showing you what the firing rate is broken down as a function of which target that the animal is about to reach to. So again, this is during the delay period. The animal hasn't moved yet, but he's planning a reach to to a certain target direction, and we can measure the firing rate during that delayed period. And here, what's plotted on the x-axis is the target direction, and then in color, again, is the reward condition. So this is how reward is modulating those curves. All right. And for our next way of visualizing activity in this high-dimensional space is we use linear discriminant analysis to find a two-dimensional plane that explained target separability. So we wanted to look at this planning dimension, the how neural activity separates different reach conditions, ignoring the reward condition. And we can find a 2D plane that best separates the different reward conditions. And to give you a feel for what that looked like, so I'm going to show you a couple of movies. Hopefully they translate well over Zoom. Here's what that looks like in static conditions. So we can find two dimensions that separate the different reward conditions well. So the different, not reward conditions, the different reach targets well. All right. So this is what this looks like. We're recording from some 90 to 100 neurons, and we can sweep time from target onset on through, and we can look at the neural activity projected into this target separation plane. And so I'm going to show you that here. And as time sweeps along, we see the activity starts in the center of this plane and it moves to the edge. And this is a single trial from one of the reach conditions. And then I'm going to show you a single trial from a different reach condition. And here's the different reach condition. You can see for this different reach condition, even though by eye, it's not easy to see how the firing patterns of these 100 neurons have changed, we can see in this particular linear combination of these 100 neurons, we see that the neural activity is moving in this other direction for this different reach condition. And in fact, we can do this for all of the targets. And what we see is that as the go cue turns on, the neural activity separates in a target dimension dependent fashion and then moves to the edge in this neural projection. All right. So that is our visualization of these preparatory signals in motor cortex. We show the animal the target that indicates the direction they need to reach. They know they have to reach quickly and accurately, so they plan a movement. That plan is reflected in the neural activity that we're recording, and it's reflected as neural activity moving to these different locations along this plane. All right. So we can look at this for each reward condition. So I'm going to start here with a small reward condition. And what I'm showing here, each dot is a single trial for the small reward condition for the target that's located. And we can show it for each of the target conditions along this plane, and we get this nice separation by target. And when we look at these single trial conditions, it's a little bit noisy. So I'm going to show you the average for each target condition separately as this ring. And the ring is connected by a red line that indicates that each of these target-dependent activity means was from the small reward condition. And the question is, what happens to these motor commands as we change reward? So we can construct this analysis again for the medium and the large condition. Let me see here. Oh, there we go. Here it is for the small condition blown up. And here it is for the medium and large reward condition. And what you'll see is that these rings expand, right? The whole size of the ring increases a little bit. And then for jackpot rewards, when we construct these rings, they collapse back down. So when we look in this plane, and this plane was found by distinguishing the different targets, what we see is for the small rewards, they're distinguishable, but for large and medium rewards, they're more distinguishable. Those means separate from one another so that the planning signals are more distinguishable from one direction to another. And then for jackpot rewards, those planning signals collapse back together, suggesting that there's less information about the upcoming reach conveyed by this neural population. All right. So I've shown you sort of 3 dimensions of neural activity so far. We've got this 2 dimensions here that represent the linear combination that best separates target direction. And earlier, I showed you a single axis that best captured the variance that was explained by reward. How do these 3 dimensions relate to one another? And it turns out that the reward is orthogonal to the target encoding. So when each of these are axes in this 90 or 100 dimensional space, and it turns out they end up being orthogonal axes. So the full picture in this 3-dimensional space is like so. Reward's primary effect is monotonic. it increases the projection along this reward axis from small to medium to large to jackpot. As it does so, the target encoding interacts with that reward signal such that the small rings start out not super well separated. They separate for the medium and large rings, and then they collapse back together for those jackpot rings. So putting all of this together suggests that there's something about the reward signal that interacting with these target planning signals that first causes an improvement in the fidelity of the reach and improves success rates, and then a decrease in the fidelity of the reach, leading to decreased success rates. And so a question we had is, or a hypothesis we formulated is that there is some kind of optimal zone, an optimal preparatory place where you can have neural activity that's going to lead to a really good reach. But if neural activity isn't in that optimal zone, the reach isn't going to be as good. And the idea here is that as your reward signal sweeps, as your reward goes from small to medium to large to jackpot, it pushes neural activity clouds into sort of this optimal zone for motor preparation, and then pushes it too far and it falls back out of this zone. All right. And so on the target projection plane, that optimal zone would look like further out on these. The farther those rings are separated, maybe the better the reach planning is. So maybe the yellow and blue points on these curves represent better planned reaches, and the red and the black points, the small and jackpot rewards, represent plans that aren't as crisp. And so we wanted to test this hypothesis, and the way we did this was we took a single direction condition, and we looked from the average of all the reward conditions on an axis that points outwards. And we called that the target preparation axis. And we wanted to look at the single preparation, so single trials, where they were along this axis. So in an individual reach, is it farther out from the center, or is it more collapsed in towards the center? Right. So those are the single trial preparatory states here that we project onto this target preparation axis. And when we project all of the different single trials from the different reward conditions onto this target preparation axis, what we see is we see these now non-monotonic curves. They go up from small to medium to large, and then they collapse back down to jackpot. All right. So now we have a function of neural activity that's showing this non-monotonic reward relationship. But that hasn't proved that this optimal zone hypothesis is true. To really get at whether this is true, what we want to see is within a single reward condition, is it true that if you're farther out, you get a better reach, and if you're closer in, you get a worse reach? So we focused in on just the large reward condition for now. And the question here becomes, if we project along that target preparation axis, is it true that the farther out you are along that axis, the better that reach is? Or if you're closer to the center, is your reach really worse within this large reward condition? And so we did that, and we said, all right, in the preparatory period, before you've succeeded or failed, can we predict whether you will succeed or fail based on how far away from the center you are? And what we found is that for this reward condition for this monkey, sure enough, you're going to succeed if you're farther out. And for failure conditions, that neural activity tends to be more collapsed back in towards the center. We ran this for each of the reward conditions for each of the monkeys, and we see this statistically significant effect where the further out you are, the more successful you are. The closer into the center you are, it really looks like the less prepared that reach is, and it's more likely to fail. Now, this is for all types of failures, but remember, jackpot rewards tended to be accompanied by undershoot failures. And so we reran this analysis just looking at successes versus undershoots. And for that, the effect becomes even more clear, where the further out you are along this planning axis, the more likely you are to be successful. The closer you are, the more likely you are to undershoot that reach. All right. So to summarize the neural mechanisms part of this tack, we found that the main effect of reward on neural activity in motor cortex is monotonic. If you're an individual neuron and reward causes you to increase your firing rate from small to large, jackpot rewards will increase it even more. if reward causes you to decrease your firing rate from small to large, jackpot rewards cause you to decrease it even more. So the main effect is monotonic. However, we see this interaction with the motor command signals such that as reward increases, you get more information about the upcoming reach in those planning signals. And then for jackpot reward, you collapse back in again as if there was an optimal zone of motor preparation and reward is pushing you into it and then past it. And finally, we saw that the further out you are along those target preparatory axes, the more likely you were to succeed. And in fact, on individual trials, we could predict if there was going to be success or failure or undershoot failure. All right, so that's neural mechanisms. So jackpot rewards interfere with motor preparation in a manner correlated with failure, constituting a potential neural basis of that choking under pressure phenomenon. All right, I wanted to spend the last part of this talk talking about implications. What do we learn from this and what do we take away from this? So the first thing I want to talk about are brain-computer interfaces. That's where the majority of my research to date has been, is understanding for paralyzed patients, can we record neural activity in primary motor cortex and then use that to control devices, cursors on computer screens or robotic arms? All right. And the implications for brain-computer interfaces aren't great. Reward information modulates motor information in motor cortex, and it has a strong modulation of that motor information. And none of the current designs of brain-computer interfaces take this into account. Nobody looks at how motivation is actually impacting that. And so this complicates BCI design, especially since it has this sort of nonlinear interaction with the encoding signals. So from a BCI design standpoint, unaccounted for motivation acts as a nuisance variable, and maybe we should design decoding algorithms that factor it out. On the other hand, it's worth considering whether this is a feature of the motor system or a bug. So on one standpoint, right, you might say motivation shouldn't matter. If you want to execute a reach, you should execute the reach. All right. And so from that sense, it's a bug, right? The fact that motivation modulates the fidelity of these motor commands is maybe something that we want to get rid of when we design our brain-computer interfaces. On the other hand, it's in the system. Presumably it serves a purpose, and it's worth considering what that purpose might be. So we don't yet know why these the motor command fidelity interacts with motivation in this way. And it'd be interesting to try and understand from a sort of normative perspective why that might be. Are there effort discounting factors taken into account that motor cortex is involved in sort of those neural computations? We don't understand yet what the purpose of those motivational signals in motor cortex is. All right, so that's implication number one on brain-computer interfaces. Implication number two, we can infer motivation from neural activity, right? We see that neural activity, that reward causes this monotonic drive signal that we can read out of neural activity, which means that we have an insight into the person for a BCI, human subject, or the animal's motivational state at that given point in time. All right, so one question might be, well, nobody wants to choke under pressure. And if we have access to your sort of underlying motivational state, can we prevent choking under pressure? Can we tell an individual, it's not time yet, you shouldn't try and execute that reach yet, you're not mentally prepared, you're not in your optimal state? So couldn't we somehow use the fact that we can infer motivation to help improve or regulate our motor control abilities? So that's one of the things that we're super interested in trying to pursue. Lastly, from a modeling perspective, this is highly underexplored. All right, we see that rewards modulate neural activity. They modulate the motor command signals, and they modulate the actual movements that you perform. None of the current models of movement that I've stumbled across so far in the literature have any kind of motivational factors built into them. And so it's a really interesting question on how they should be built into them. How should these motivational signals be put into recurrent neural network models that drive, models of the arm? How should we expect neural activity to respond to these? What does it mean about the training signals that we use? So from a modeling perspective, how can we build motivation into either neural or biomechanical models of motor control? So that's a big open question so far. Finally, I've been using the word we a lot in this presentation. I want to talk about the people who really make this work possible. And so here's a list of collaborators and people in the Chase Lab and funding sources. In specific, I want to highlight in red are the people who have worked on the reward projects over the past 8 years or so. And in maroon with underlines are the people who have collaborated mostly on the stuff that I've mentioned today. And so that would be Aaron Batista, who's my longtime collaborator. He's faculty at University of Pittsburgh, and Adam Smolder, who was the graduate student who spearheaded most of this work. So yeah, that is the end of my talk, and I'm happy to take any questions that you might have.
Thank you so much. It's a wonderful project, and we have learned a lot. I think anyone from the audience want to ask any questions?
I got one. Thanks so much for the talk. This was fascinating. I'm just curious how much of what is the gap between, let's just say, like studying this in under controlled setting versus what happens in the wild setting. Like, do I take the lessons from here and then put it in players who are playing NFL and Olympic level sports? What kind of gap are we looking at?
Yeah, that is a fantastic question. And I don't know that we know the answer to that very well, right? When we study things in the lab, it always ends up in these controlled scenarios. Although I will say, the first time choking under pressure really came to bear, it's in sports, right? And many of those scenarios are quite controlled themselves. If you look at field goal kicks in football, that's relatively controlled scenario. Players always line up in the same way. The ball gets chucked, it gets held, and the kicker has to run up and kick it through the field goal. We've run studies looking at field goal percentages, controlled for distance from the field goal and weather conditions and all the things that we can control for. And field goal success percentages decrease when you're in the last 2 minutes of the game and the kick could tie or win the game. So it looks like choking under pressure in the wild, in scenarios that match the lab as closely as possible, in that they're pretty controlled scenarios in this sports scenario, it looks like very similar to what we're seeing. So I think from that standpoint, I think what we're seeing in these animals is true to what's happening in the real world. Maybe it's at a slightly, sort of slower pace scenario, but I think it's true to what's going on in the brain in the wild. When it comes to the implications and how we control for it and that sort of thing, that is a different can of worms entirely. We don't have access to the neural signals. So how do we do that in real time? I have no idea how to answer that question yet.
So there's a very strong argument, which I think I'm fairly convinced that we find ourselves in a similar situation. Like you said, but will the solutions find in this case A will transfer over to that case? I think that remains to be seen. Okay, so maybe my a little bit of a conservative version of this question is, in what scenarios have we applied these findings right now in the real world on, like, in humans? Like, do you have any examples to, like, maybe ground my understanding on what settings have we found things to be closer and applicable yet?
Yeah, so I would say right now the arrow is currently working the other direction. So in sports, people have known about choking under pressure for decades. And it's been how do we train athletes not to do this? And there are people who try and, visualize and ignore the crowd and all of that. There are coaches who will go through and work with you to try and make it so that you don't get the twisties in gymnastics and you don't get the shakes. And, and so I want to say there's a lot of use cases out there where trainers have adopted strategies to try and make you overcome this. We've only known that this exists in animals for about 3 years, 3 to 4 years. So how those scenarios translate to animals and then what we can learn from animals that's new and translate back to humans, that we don't know yet. I would say one of the interesting things that we have found that doesn't agree with what seems to be the case in humans idiosyncratically is that all of our monkeys choke and they don't train out of it ever. That doesn't appear to be the case in humans. We know in sports there are people who are absolute ice, right? They can perform the same action. It doesn't matter what the pressure is. Some athletes just get better under pressure. My favorite example is Tom Brady. When the pressure ramps up, he just gets better. So I think what we're finding in animals is that the effect seems to be much more reliable than it is in humans. To me, that indicates something interesting. We used to think that we choked under pressure because we overthought it. Our big brain got in the way and caused us to overthink it and fail. I no longer think that's true. I think our big brain actually helps us create scenarios that can partially mitigate this effect, that sometimes we can cognitively reframe the scenario so that it isn't as motivating and performance can be optimal. And monkeys have a hard time doing that. That's what I think. I haven't proved it yet, but I'm super interested in trying to follow up with it.
I think I might be taking more of my time, but I mean, it might be the case that like Tom Brady came to the selection pool after a lot of filters, and he just picked the first monkey he got.
So it could be we have a Tom Brady monkey. We just haven't seen him out there yet, right?
Perfect. Yes.
One in a million.
I'll let others go, and I have more questions. I'll come back to it.
Yeah, thanks, Vikash. Like, anyone have more questions to jump in? Jeff, feel free to jump in. I think Ping Yuan have one. Like I have a question, like in the chat, like why do monkeys fail? They usually undershoot rather than overshoot, and how would you explain from neurodynamics?
Yeah, from a neural dynamics standpoint, I would say the movement isn't as well prepared, and therefore it doesn't execute with the proper vigor. So you end up moving too slowly, you end up landing too soon, you end up with a movement that's hypometric. That's from the neural dynamic. So your preparatory state isn't quite where it needs to be to trigger the movement. And so when you go to trigger it, it takes a little bit longer. That would be my explanation from that standpoint. From a maybe psychology, cognitive standpoint, there have been a lot of theories about choking under pressure. One major theory is overarousal. The reward, signal comes in, it's a jackpot. Oh my goodness, it's a jackpot. And you're too jumpy. That doesn't seem to be supported by our data. Our animals don't seem to be too jumpy because they undershoot on these reaches. In contrast, it seems that this other thing is going on. They get in their heads, they try and be too cautious. They try and be too accurate. And because of that, they forget the time constraint and run out of time. That seems to be a more consistent explanation for our behavior, at least.
Yeah, great. Anyone have questions?
I'll go for one more.
Okay.
Oh, I think someone is asking. Like Tony?
Yeah, sure. Go ahead. I'll go ahead. Thank you, Professor Chase, for this amazing presentation. I have a very, I don't know, it's a very big question, but evolutionarily, do you have an idea why is reward information or motivation modulating movement information in the motor cortex? And I mean, is this suggesting that the motor cortex participates in value-based decision-making?
Yeah, these are excellent questions and stuff that we think about and ponder quite a bit. Why does this exist? It doesn't seem to be, from an evolutionary standpoint, doesn't seem to be optimal. I have a few answers to that. Number one is that I don't think we are optimal evolutionarily. We trend towards optimal. Every new design is hopefully better than the design that comes before it. But I don't think we're optimal yet. I don't think we're the endpoint of optimality or of evolution. So that would be answer number one. And it could be that this motivational drive really does have good reasons to exist, especially from small to large, and why movements get better from small to large reward. And one of those things is that a movement itself is costly, right? To move is to exert effort, and to put yourself in a new place, and it could be unsafe, and all of those things, especially back in evolution when we had predators we had to worry about and all of that. Moving is a dangerous endeavor, and it consumes calories, and you should perform computations to see if it's worthwhile, and reward needs to be a part of that. Is the outcome of the movement worthwhile for executing the movement at all? I have to exert more effort to execute that movement faster. So does the reward justify the extra effort to move faster? And maybe I only move faster when the reward is larger because of that. And there are good foraging theories that suggest that that is the case. Why it breaks down for jackpots? My personal theory that we haven't proved yet is that we just don't work enough on jackpot scenarios where when that reward drive comes in, it's so massive that it pushes the neural circuit out of where it's been trained. And so I think these jackpot reward signals, probably conveyed by dopamine, project into motor cortex, and they push the circuit out of the part where the synapses have been kind of optimized for movement. That's my guess. Now, does motor cortex participate in value-based decision making? I wish I knew the answer to that question. We don't yet know if it's participating in the computation or if these signals are coming in and sort of activating the circuit differently. And so jury's still out on that.
These are amazing answers. Thank you so much.
Yeah. Anyone have any more questions? I think we should have tons to ask also. Yeah.
I'll ask one last and then I'll shut up. What will be an ideal experiment would you like to run?
Yeah. One of the things that we're trying to do right now that we haven't gotten off the ground yet is we want to record from dopaminergic neurons. So the reward processing centers of the brain. Reward is processed by dopamine neurons, mostly in the substantia nigra and the ventral tegmental area. And so what we'd like to do is record from these dopamine centers and find out what's happening with the reward signal itself. Does the reward signal cause choking? Does it know about choking? So in other words, on jackpot rewards where you're going to choke, is it abnormally large or abnormally small, or is that something that's coming afterwards in a different part of the circuit? So that's one of the experiments that we'd love to run. I'll give you two. Another experiment that we have started and that we do have some results on is taking this out of the movement domain into other domains. So it turns out that motivation affects not just how we move, it affects how we perceive and how we decide. And so we've been starting to look at tasks that have those features as well. So change detection tasks where you're looking at a stimulus and you have to decide, is that stimulus the same? So that flashes on and off. And when it comes on one of those flashes randomly, it's going to change and you have to decide which one. And if that movement, if the change in the stimulus is small enough, that can be a hard task. And we can run that under a reward scenario and see how does reward affect perception? And is it the same kind of overall mechanisms maybe playing out in a different brain area? So that's the kind of thing we're working on these days.
And do you think like our reward mechanism is pre-programmed into our DNA, or we train our reward mechanisms in addition to training ourselves?
Yeah, I think it's a combination of both. I think that the fact that dopamine signals are stronger for bigger rewards, that's hardwired into our DNA. How we act in response to those signals, I think, is very much a nurture and training scenario. So yeah, I think it's very much a combination of both.
Have you finished your questions? Yeah,
I have 10 more, but I think in the interest of time, I'll just wait.
Yeah, I have one technical ones. Like when you are talking about the reward axis and the preparational axis, when you are dealing with the like high dimensional space, right? Would you think there would be an artifact of some kind of dimensionality that you find? For example, you are always going to find the reward axis, but you're always going to find the non-reward axis, right? Like given that you have like 400, 500 dimensions, and then you're going to always find the preparation like axis, which like you can imagine in that space, you can also find the opposite one. So like I was just wondering, would that be an artifact, something like that? How do you
Yeah.
like?
It's a great question, right? How do we, because you're right, when you're in high dimensional spaces, right? Small signals can cause you to, small amounts of noise can cause you to misinfer what's happening. So we take a number of different approaches to mitigating that. The first is that all of the axes that we find are cross-validated, right? So that they're predicted on data that wasn't used to find them. And we find that's the case again and again. We find that the axes are repeatable across monkeys, right? So if we go through this procedure in each animal, we tend to find the effect. We make sure that all the predictions, like the success and fail cases, success and failure wasn't used when we define the axis, right? So that is a consequence and not something that's baked into the design. And finally, in order to get enough data to really be solid about these results, we combine data across multiple days. And so we've actually conducted, and this is part of the brain-computer interface design work that we've been doing for a long time, is we've developed methods of stitching recordings across time into a common low-dimensional space. And we've figured out how to align those spaces. And we did that initially so that we could stabilize BCI decoders so that they wouldn't have to be calibrated every day. But it turns out for studies like this, it's really good because we can combine thousands of trials over a month of recording and get tons of data so that when we're estimating axes in this space, we can do it with high confidence.
Do you find, for example, like two reward axes like that's orthogonal to each other? Like that's
Yeah. So when we find these reward axes, we're doing it based on 4 points, right? We've got 4 reward conditions. We average everything. We take the marginal across all the other conditions, and then we get a small reward, medium reward, large reward, jackpot reward, right? So it's 4 conditions. The first axis we find explains the most variance of those 4 points in this high dimensional space. There's always a second axis. It tends to be, non-monotonic. And it tends to also predict success versus failure. And we haven't, so it's there in sort of the smaller variance components, and we haven't figured out how to interpret those yet as easily as with the other factors that we found.
I see. So you do find like second, like third, or fourth.
Absolutely, yeah. But the primary one explains, I think it's over 90% of the variance. The second one explains 4 to 5% of variance.
Yeah, that's my question. Thank you. Lukash, you have more? No? Okay. I think yeah, it's been thank you so much for joining.